Hello, hello. How's everybody doing? I hope everybody's doing great. With the outbreak of the coronavirus, I've seen a lot of people I've made a lot of videos about it, but none of them is like really looking at the basic science behind it. So I felt the need to, just make a little video and kind of talk about the science behind it. I hope everybody is trying to stay positive and practicing all the good hygiene stuff just to make sure that they don't get the coronavirus. I hope what I want to tell people is that it's like everything come and go. This one will also come and go. So we just gotta sit tight and wait for that moment. I think we got a CDC and other people working on it, so I think things will get better. Okay, with that being said, I wanted to explain the background of what it is and what the coronavirus is and how it works and stuff like that. But before that, I want us to go to the basics, which is what is a virus? So a virus is a biological agent that reproduces inside a living host. So what I want you to take out of this is one, virus is a biological agent, which means it's not a living thing. You know what I'm saying? It does not have its own nucleus and stuff. It just have genetic material, but it cannot use that on its own unless it gets a host and use the host machinery to reproduce. Without a host, it's useless. So that's why it's what acellular, because it's not a cell. It's not a living thing. It is only active when it's like the host, and it uses the host stuff like machinery and stuff. But it doesn't have its own. So we got two types of viruses. We have different types of viruses, but when you're trying to compare in terms of like envelope or no envelope, then we got two types, which is the naked one and the envelope one. So the envelope one have a big envelope around it, made up of glycoproteins. And we have the naked one without the envelope around it. The coronavirus is an envelope virus, which is kind of a good thing, to be honest, because most vaccines and drugs normally target the envelope around the virus and curing it. So it's good to know that it has one. Yep. Another word also that people have been throwing around is virion, which I wanted to just explain it. Most people use it interchangeably with virus. At the end of the day, it kind of means the same thing, but what virion is a complete functioning virus outside of the host. So a complete functioning virus, a virus that has, the virus has gone inside the host, replicated, and formed new viruses. And this virus is just gotten out of the host and is ready to attack. It's outside of the host and it's just ready to attack the next cell. That is what a virion is. Yep. So just like I said, we have a lot of types of DNA, but in terms of envelopes or the envelopes around it, we have the naked and the envelope virus. We also have different types of viruses in terms of its genetic material. We have some with DNAs and some with RNA, some with positive RNA and some with a negative RNA. And there's a whole lot, but we don't have one that have both. But yeah, they have some sort of a DNA or some sort of an RNA in a capsid. And the capsid is like that quote that's around the DNA or the RNA. That's what we call a capsid. It's a protein coat. So now we have the star of the show. What is a coronavirus? So first thing first, it is a virus, obviously. But what kind of virus is it? It's a single-stranded positive RNA virus. Single-stranded positive RNA virus. Okay, so what this means, what I want you to take out of this is positive RNA virus. When we say positive RNA, all it means is that it can make proteins without undergoing transcription and can come from RNA translation that make proteins. In order for most living stuff like humans to make proteins, you have to go from DNA to mRNA. Then we can get the proteins. But escape that, because it's a positive RNA, it skips that middle part and goes straight to making proteins. It is envelope, as I said, it is an envelope virus. It has that envelope around it, which is made up of glycoproteins. So they say the exterior parts or spikes are made up of different types of glycoproteins. Yep, and we have you have the S, E, M, and N glycoproteins. These are just indicative of the various structures on the envelope. So there's the spike, the envelope, the membrane. The indicative of the structures of the virus. So the spike, the envelope, membrane, and nucleoplasmid, which is the RNA and a bunch of proteins inside the core. Yep. So on the news, we hear that this new virus this the novel corona, I don't know, COVID-19 or whatever you call it, which is true. It is a new strain of coronavirus, but it's not a newer coronavirus. Coronaviruses have been existing for a long time. They've had various coronavirus affecting animals. I mean, it is believed that this coronavirus came as a result of, some people feasting on animals. But and it's also not the first human coronavirus. Actually, the first human coronavirus was isolated in 1966 by somebody who had like a cough problem, because it's a respiratory virus. So it kind of affects the respiratory part of the living organism. Not all of it though, but this virus, the one we're dealing with right now, affects your respiratory parts. Yep. And one thing it does is it has tropism. So when I say tropism, it's just basically it is selective in which organ it attacks. That's why you don't have coronavirus. It doesn't attack your feet and make your feet hurt, but it attacks your lungs and stuff like that, your respiratory tracts and stuff like that. Yeah, that is where it attacks, and it's also species specific. It just doesn't attack like anybody. There's for example, just because this virus can attack a human being doesn't mean it can attack a dog. Because it's really specific. On the person or whatever organism it attacks. Yep. So there's kind of a diagram that shows the life cycle of the coronavirus. This is actually that of the SARS, which was big in like 2003. But this coronavirus has the same pathway. So yeah, I wanted us to look at it a little bit. So first we have, fully functioning virion, is active, is all filled up, it's ready to attack. It is a virion. So first thing it does is that the spikes around it, that was what makes it specific. That's what attaches itself to where it has to be. And the receptor it attaches to is the ACE receptor, was the angiotensin converting enzyme receptor. And fun fact, it's actually the same receptor that the SARS virus back in 2003, that was the same receptor it was also attaching to. So that's kind of like the docking point of the virus. So they all have the same docking point. So once it has a docking point, it has various ways which it goes into the membrane of the host. It can just fuse with the host membrane and just gets inside just like that. Or it can, form a butt around it. Something called a vesicle is like it's inside a bubble and it comes inside the cytoplasm of the host cell. This process, which is known as endocytosis. So yeah, when it gets inside the cytoplasm, that's where it likes to work. It's offloaded, its RNA, releases RNA, and what it does straight away is, I told you that it skips one process during protein formation. So what it does after it unloads is start making this protein known as replicates. And it breaks it down so it becomes a bunch of them. And what these replicates do is just replicate the RNA strand. It just replicates it. Yep, and it forms a negative RNA. Remember, we started a positive RNA. But when you make it makes the complementary pair. So it makes, which is the negative version. That's because that's what complement to that positive. So it makes the negative version. And with the negative RNA, it cannot skip the transcription process like the positive does. So it goes through the right channels and make the various proteins needed for the new viruses it's making. And it also use some of them. Yep, it also keeps, some of the RNA for the inner genetic material. And after it is mature and ready, it kind of leaves the host cell. So it kind of leaves the host cell. So yeah, so I already looked at that. I already talked about tropism and the receptor. Yep, so another thing we have to know about this virus is it is a non-lytic exocytosis. So when you talk about non-lytic, whenever you hear the word lytic, I mean splitting up or just basting it up, just like you just bast like a balloon or something. That's what most viruses do. I mean, not most viruses, but most like really violent viruses, after they're done, multiplying, they just slice the cell and just rush out of it and just split the cell open. But this one does not do that. It's a non-lytic exocytosis, so it doesn't leave in a really aggressive fashion and splits the whole cell open. Yep. So another thing that's really heavy in the news is testing. People be complaining there's not enough test kits and there's not enough labs that can test it and a whole lot. So I wanted to, talk about it a little bit and see what tests they actually doing. So the test they use to check whether they have the coronavirus or not is based on PCR, which is polymerase chain reaction, which is a technique used in making many copies of a specific DNA in vitro. So basically, if you have a specific DNA, if you want to multiply it and make a lot of it, we use PCR and do that. And when we say in vitro, it means that it is done outside the whole cell. That's why the test, I think you got to swap. They take your DNA and they just do the test and they test you. They don't just do it like in you because it's in vitro. And PCR is a really old technique. It's used a lot in most labs. Colleges. I'm a college student, and yeah, we use it almost all the time in our various lab classes. So it was a really trusted, process. So I just wanted to talk a little bit about it. It has to make PCR has three main processes. It has the denaturing stage, annealing stage, and the elongation stage. But before we go through all that, first it needs components. So how was it detecting it? So what they do is what a tested kit have is called DNA primers. So DNA primers are like the police. Okay, it's gonna go into the DNA and detect the part where the part of the gene that has the coronavirus or the corona genome. It's gonna attach to that part because that's how it is specifically designed to do. And the structure known as DNA polymerase, which is more like the builder, that's where it attaches and start to build more DNA using something called nucleotides, which are like the building blocks. Okay, so the primer is like the snitch. It's gonna point out where the virus is, so they can just make multiple copies of its genome, so it can be expressed, and scientists will know that, oh shit, there's a lot. This person has it or this person doesn't. So what it does is for the denaturing stage, it splits the DNA. The annealing stage, it attaches the primers. The primers attached to close to where the coronavirus genetic material is. And the elongation, it grows. And after it grows, they'll have a way of detecting it. I'll think by using gel electrophoresis or any other technique to detect whether you have it. But the main process is if you grow a lot, that means you have it. Because if you do not have it, the primer cannot, has nothing to point at. You know what I'm saying? If you do not have it, the primer has nothing to point at, so there will not be any large growth. But if you have it, the primer has something to point at, so there's going to be a lot of growth. You're going to know you have it. So yeah, that's the pro for PCR. It's a trusted process, been around for a long time. The con is sometimes they can be a false positive. So the primer, because a lot of primers are added, the primer might, instead of detecting the coronavirus genome, it might detect its fellow primer as the genome, which is a false positive. So when the test comes, it might seem that you have it, but the primer is just finding another primer instead of finding the corona strain. So I think what the CDC test does is they add more primers. So you have, because the false positive it hardly occurs. So you cannot form like a whole, you cannot really amplify the DNA if you have a false positive. So if it comes back and you have a really large amplification, it means you really got it. But you have a really low amplification means it was just false positive. But in rare cases, somebody might kind of be in the middle. I think the ship in California, one person was kind of undecided. I think that was what happened. Was kind of in the middle. And if that happened, I think if they retest you, the truth shall come out. Yep, so ways of stopping the spread, just try to maintain social distance, 3 feet. I read somewhere that's now 6 feet, so just everybody just don't go into other people's personal spaces and try to prevent other people from coming into your personal space. Avoid touching face because you have a lot of, holes in your face, your mouth, your nose, your ears, so places where the viruses can go. So just avoid touching it and wash your hands frequently. Try to practice hygiene and respiratory hygiene. Maybe if you're coughing, make sure you don't cough, in the midst of people. If you think you have a runny nose or something, you have to wear a mask or something because, the mask doesn't protect you if you don't already have it. But if you already have it, kind of lowers, the chances of somebody close to you getting it. So the symptoms are fever, cough, difficulty breathing. So regular respiratory symptoms. So it's nothing outside the box. So I think that's kind of what makes it hard because it gets confused with the flu sometimes. But yep. Yeah, but I think if you're having these symptoms, then you probably need to see your doctor for help. Yep. So people, that is it. I hope everybody stay positive and stay strong. And yeah, if you think I probably said something that was a mistake or something, you can just put it into the comment section so we all can learn. And okay, goodbye.